Hi, everybody. My name is Benjamin Walters. I'm a senior scientist at Genentech in the Department of Biochemical and Cellular Pharmacology. I'd like to thank Thermo for inviting me to give you this seminar today, where I get to share with you a little bit of my experience so far at Genentech using hydrogen exchange. So I thought I'd begin by telling you a little bit about how I got into this, because I didn't actually think that I was going to become, an expert in protein hydrogen exchange when I started graduate school. Actually, I went to graduate school. I was interested in how does a protein fold? How does it do it in a biologically relevant time scale? There were these two theories, one about a protein folding funnel and another about this pathway theory that had actually existed for a long time. And the problem was there wasn't really any good technologies to measure this with the level of resolution needed to say something about how the process worked. I ended up going to the lab of Dr. Walter Englander that you see over here on the right, and I quickly learned that if I wanted to study protein folding experimentally, I was going to need to learn about a technology called hydrogen exchange, or amide hydrogen exchange. One of my favorite quotes from Walter was that you can learn everything you ever wanted to know and everything you didn't about protein folding with hydrogen exchange. But activity in a protein doesn't come from a rock. It comes from the movement in the protein. And so we need to understand these things in order to understand protein activity. If you're more interested in protein folding, that's not really the focus of my talk today. You're more than welcome to have a look at this paper. It's actually a really good paper that summarizes a lot of Walter's work on protein folding. It was published in PNAS in 2014. I'll briefly tell you a little bit about at least my takeaways from working with Walter and this really fun experiment. I'm going to kind of walk into hydrogen exchange first, starting out very conceptually and telling you about an experiment called kinetic pulse labeling. In this experiment, basically you can think of it as we start with the protein that's unfolded and we dilute it into folding conditions. We have this stuff, HX spray paint, and when we spray it on the system for a very small amount of time at a certain time point, anything that's folded resists getting painted. Anything that's not folded gets painted, okay? And then we can chop the protein up into little bits and we can look at what got painted and what didn't get painted. And we can say, hey, was this piece folded when I did the pulse or not? In doing so, we can learn a little bit about the order of events in protein folding. And actually in my dissertation, these are kind of the main points of a lot of work in my dissertation. I was trying to understand how this protein, maltose binding protein, actually made its way to the native state. Some of the important raw data is being shown over here on the right, where this is folding time, the time until I gave it the pulse, and the fraction of the population that didn't get painted. And what you can see here is that there's a fraction that seems to resist getting painted a lot earlier than the others. As it turned out, that was because the protein formed an intermediate within about 9 seconds. And then it took some time to shake out what we think of as potentially misfolded states or something that happened in this stage called a collapse, which I'm not going to talk a lot about right now. But once it started to shake that out, it could then fold this little green piece and then proceed to fold the gray and the red in time. This explains how proteins fold in a biologically relevant timescale. They do it because they don't all have to fold at once. They fold in subglobal units. It was really interesting to note that there are 3 mutations known in maltose binding protein, shown here, that prevent the protein from effectively folding. And if a pathway hypothesis is true, this makes a whole lot of sense, because if you can't get through this intermediate, you can't make it to the native state. It stands in contrast to folding funnels. And the only way that you can get this information is by an experiment like hydrogen exchange, which can show you that at 9 seconds, certain pieces are folded and other pieces definitively are not. How did that experiment work? Well, we have to look a little bit at the fundamentals of hydrogen exchange in order to see how this works. This is a really old way of writing kind of the hydrogen exchange reaction, and we're going to focus on the amide. The reason that we're going to focus on the amide is because it exchanges at just the right rate when it's exposed to water. So all of the other protons in the protein are exchanging, but they're either going too fast or too slow. The amide exchanges at just the right rate. And what I mean by that is this chemical exchange rate. So we can look at the chemistry here. The chemistry depends heavily on pH. It depends on the donor pKa, which is in which is affected by the sequence, and it depends on the temperature. You see down here with pH, we have a minimum at about pH 2.5. This is really great because it allows us to effectively slow down the exchange reaction so that we can measure it. We can slow it down to about 1% per minute. Or if we get up to, say, pH 5, 6, 7, 8, even into the higher range, the exchange drops down to the milliseconds time scale. And so you can measure everything from very slow opening reactions to things that are actually quite fast by just simply modifying the pH. Now, why does it tell us about protein dynamics? How is this a protein dynamics experiment? Well, as it turns out, that's because there's structural physics involved. When there's a hydrogen bond to the proton, it cannot exchange. It is precluded. It is protected. And so the only time that it's available to exchange, the only time that it's available to go at this rate constant, depends on the structural physics of the opening and closing reactions. And sure, if you're, it tends to be the case that amides that are exposed to solvent spend less time in hydrogen bonds than amides who are buried in the center of a protein. And this has given rise to a level of solvent accessibility. I like to think about it as hydrogen bonds. Now, how we actually do the experiment, how we actually carry it out, and this experiment's a little bit different than the spray painting experiment, but fundamentally it would be done the same way after the quench step. In the native state experiment, we have a folded protein that we just diluted into D2O. This is the experiment that you'll be probably more familiar with too. And then we take this protein that's diluted into D2O, and we watch it slowly as it exchanges, as all of these amides go from H to D. And they're dependent on the stability of the structure or the stability of the hydrogen bonds. By dropping the pH or quenching, we can then digest the protein after we've labeled it, separate its peptides, and then measure how much mass they've taken on with respect to time. So this is done after we've done the labeling experiment. And then by piecing together all of the peptides that we've measured, we can see that structure or a significant standoff from the kinetic exchange rate is observed as we start to get into residues 11, 12, 13, 14, 15, 16, but not before. So residues 1 to 10 don't show us much of an effect. And in this way, we can start to see, okay, stable structure exists in these sites. Conceptually, that experiment is working like this. We have the protein, we put it into D2O. Slowly, all of these black spots, which represent hydrogens, are going to go and exchange themselves for deuterium. And then we're going to be able to see where it is by chopping the protein up into different peptides. We can look at the thermodynamics by looking at the standoff. How long did it take to see the equivalent impulse on any given peptide? That's going to tell us something about the thermodynamics. And this paper really goes into detail about that. I'd recommend it if you're interested in the structural biophysics of this experiment. But an experiment that you've seen done very often is epitope mapping, where we're basically adding a binding partner and looking at the perturbation effect that the binding partner had. I just want to stress, though, that everything in native HX is kind of a comparison experiment. Either we're comparing how long it took to exchange between the chemical exchange rate, our theory, and what we observed in the experiment to tell us about structural thermodynamics, or we're looking at the difference in time to exchange. So in a peptide that's affected by the binding partner, it might change the exchange trace from, say, the blue line to the green line, or maybe from the green line to the black line. I wanted to show you one quick example that you can read more about. It kind of wraps everything together on the native state end, where we were actually able to show that the affinity difference between a wild type antibody and what's called the YTE mutant was driven by the structural thermodynamics of the two proteins before they bound their receptor. So how does that whole thing work? Well, basically this is the receptor is called the FcRn receptor. And it's really neat because the affinity of the antibody for FcRn only exists at low pH. At high pH, it has no affinity. When you looked at the crystal structures of the YT mutant and the wild type bound to FcRn, there was really no discernible differences between them. The sites of mutation weren't really making any extra contacts with FcRn. And then if you looked at the crystal structures in the unbound state, you saw a very similar thing. They're basically superimposable. There's no real differences. So you couldn't explain using structure why did the YTE mutant have higher affinity for FcRn. And so it turned out that, that the affinity for FcRn of the YTE mutant was really driven by the destabilization that occurred and was compensated for by binding to FcRn at low pH. If you'd like to read more about this story, we summarized it in JBC a few years back, but it's a really good example about how structural thermodynamics can be used even in epitope mapping experiments. But you might be asking at this point, well, how does protein dynamics impact industry? I mean, a lot of this seems like basic science so far. I mean, it's great to understand all this stuff about the YTE mutant, but I think the salient message there was that we needed dynamics to really understand the picture. And I think, as it turns out, protein dynamics really leads to all the emergent activity that we see in the lab, whether it's aggregation, which is affecting shelf life of our products, or whether it's the mechanism of action. Why is it that this compound binding in a certain location is silencing this protein? Or why is it that when we made this one change to this compound, it stopped binding? It doesn't make a whole lot of sense to me. Well, a lot of this is because of the dynamics of the protein, and we're missing that picture if we're just focusing on the structure. So, I called this presentation Running the Gamut because I've fortunately, been at Genentech for a little while, and I've had the opportunity to work on a lot of different problems throughout early stage and late stage research and discovery. So I thought I'd tell you a selection of 3 of these problems today, and try to proverbially run the gamut here, and tell you at least a selection of one from each side of the game, research and development. And so I'll start actually in early research, where a project team has a target and they're just looking for a foothold. They're looking for a way to begin. We actually screened a much larger set of compounds, but I'm showing you up to maybe 10 here. 4 compound results are being shown here shaded onto the protein. The red shading is showing regions where we saw differences when we compared the 2 experiments, bound and unbound. That all of these compounds are sharing a similar region, this little area here. But then they're all also showing us some unique signatures as well. And so we are getting, like this one is having a very large allosteric effect. Maybe that's important, maybe it's not. Can we distinguish between allosteric changes and maybe a second site binding? Well, what's interesting in what this plot shows us here is that if the changes that we're seeing on the protein are induced by a single binding site, as we titrate through the KD of that binding site, all of these changes should move together gradually back to the unbound state. And so actually that's exactly what you see in peptides 82 to 97 or even 168 to 174. I mean, regardless of the magnitudes of the change, the shift between unbound and fully bound happening at about the same rate at every single peptide. That tells us that it's allosteric. We can also increase the throughput by doing a selected time point. Now, we don't know a priori which time point we need to sample, but what we learned by doing an initial set of experiments was that in these 2 peptides here, which are affected by every single compound that we tested, if we sample for 100 minutes, we're going to kind of get the maximum of this effect. And so the idea then would be that we could then screen through a large number of compounds, sampling with a single time point. And then we could bend compounds with similar mechanisms of action as deduced by the effects that we saw by HX. And then another added advantage, getting back to some of the thermodynamics I was just talking about, because you remember, it's not just basic science. There's some relationship between the energetic consequences of binding, the protection factor that we talked about, and the affinity of the compound. As you can see here, compounds with higher affinity tend to produce larger protection factors. This is very interesting and potentially very useful in the future because you could screen on the basis of protection factors. It's also useful because you will expect to find the largest protection factor at the site of interaction. And so using protection factors, sometimes you can also try to kind of separate out between allosteric change and direct effect. To summarize this experiment, the small molecule hit validation, we gain speed with reduced ambiguity. And that's really valuable because, information is critical to making good decisions, especially in early stage research. Being able to see the difference between a non-specific binder and something that's binding specifically to a location is very meaningful because it could allow us to pursue the correct lead molecule. It also can allow us to start understanding the molecular attributes of the system and connect the target design profile to that. As we move into the later stages of research, things like mechanisms of action become much more important. Really being able to detail the epitope becomes important for, say, legal filings. So I'm going to tell you a really fun story that spanned a couple of years here, where the quest was to inhibit beta tryptase, and I got to work with a very talented team of individuals. Henry Mohn was the lead author on both of these papers that summarize our work. But you know, I would say that hydrogen exchange here not only led to an epitope differentiation, but I think it led to structural information because it allowed us to generate hypotheses and then test them in the lab. You know, briefly, the way that beta tryptase works, it's involved in asthma. It's a lot like trypsin, a little bit different. In trypsin, you start off as a zymogen. Cleavage allows for pro-domain insertion, which leads to activation of trypsin, as you can see here. And that's really just because you've changed the orientation of just a few residues here in the active site, the catalytic triad. Okay, now if we take this model and think about it in the context of tryptase, it's very similar. We have to have the pro-domain inserted, but there's kind of some extra pieces over here on tryptase. And in order to stabilize and so that this active site can become active, you basically need to tetramerize, whereby these additional loopy pieces are going to reach over and interact with a neighboring subunit to create a tetramer like this, where all of the active sites point into the tetramer center. This creates kind of a therapeutic challenge because the active site can't be hit by endogenous protease inhibitors like anti-1 antitrypsin, simply because the active site's not accessible. This project team ended up with a few lead molecules. 3 are being shown here, and I'm going to focus on 2 of them. The plot to the right shows you the effects of inhibiting tryptase activity with the full-length antibody, or IgG, which is shown depicted down at the bottom. It has an Fc domain and 2 Fab domains with a hinge that connects the Fabs to the Fc, or as Fabs only, so meaning we can clip them at the hinge and release these Fabs. The 2 things that I want you to focus on here are what happens to the green versus the red trace when we clip off the Fc domain. In the case of the green antibody, it requires the full-length molecule to inhibit. When we produce the Fab, it stops working altogether. It also has this interesting thing called the hook effect, where it stops inhibiting at high concentrations. I'm not going to focus on that here. In contrast, the red antibody, 31A, it works whether or not it's a Fab or a full-length antibody. Now, this wouldn't be too puzzling in and of itself. I mean, different antibodies work in different ways. But these antibodies compete for the same epitope. So why do they have such different mechanisms of action? And then a secondary question, why are we having such a tough time with crystal formation? So the project team could not get a crystal structure in this case. They were really struggling. They came to me with some material, and I said, yeah, I'll do some hydrogen exchange here. The hydrogen exchange results are colored onto the tetramer here to the right. 31A is shown in yellow, and E104 is shown in blue. You can see why they would compete with one another. There are residues that are in common. There are also residues that are different. This blue patterning goes down here and over here. And you know, in looking at these results, we started to think about, okay, well, you know, perhaps it's simply a matter of steric clash. Maybe maybe 31A Fabs sterically clash with one another if they both bind like this, and so there would be 2 that would be coming kind of into the page down onto the subunits, and then 2 coming up from the bottom. You know, maybe they sterically clash, and that forces these things apart. And then we thought, you know, in contrast, this looks like this is a little bit more to the side of the antibody here. So maybe this antibody kind of binds a little bit more kicked over to the side. And so maybe without the hinge present, you could actually have 4 Fabs bound to the same tryptase tetramer. And then maybe in the 31A case, you couldn't do that. And that's why 31A led to dissociation as a Fab and not E104. But then, you know, as a quick aside before I tell you the answer to this question. Why did one work as a fab and one not? There was always this background question: can any of this information help us figure out how to get a crystal? Well, as it turned out, it did. And I'm giving you this slide. This is directly from Henry, who is the crystallographer on this team. And he tells me that the hydrogen exchange results directly led to him figuring out how to do this. So the green and blue shaded regions show you residues that were affected by binding of the antibody in the presence of 31A. The green is showing you the ones that were consistent with the crystal structure. So all the regions that were seen by HX and then also contacting in the crystal structure. Versus the blue were the allosteric changes seen by HX that were not observed in the crystal structure. Eventual. Now, so I kind of blew the story because I'm talking about the crystal structure before I'm telling you whether or not we were able to generate it. But it wouldn't be a very good story if we weren't able to get it. The whole idea that Henry had was that he was looking at the system and said, okay, well, the tetramer is required because the monomer can't be crystallized. And if we can't generate a tetramer bound to a fab because the fabs cause dissociation, we're never going to be able to get a crystal. And this is because of the dynamic instability of beta tryptase in this region. It needs to actually form this tetramer or it's very unstable. HX, though, indicated that there were no real changes in the active site, that all the changes that were due to the antibody binding were away from the active site. And so Henry thought to himself, I wonder, usually making a complex larger is bad for crystallography, but I wonder if adding a, an active site inhibitor might actually work in this situation. Maybe we could use the soybean tryptase inhibitor, or soybean trypsin inhibitor, excuse me, to bind to the active site, stabilize this region of the molecule. Maybe this will allow us to get a crystal. So in Henry's words, he never would have tried this without the hydrogen exchange data, suggesting that there would be no competition between the STI and the fab. And in any event, he attributes the fact that he saw this HX data to being able to generate this crystal structure, which was incredibly useful because this became our lead candidate. Once we had the crystal structure, we were actually able to start doing some modeling to see that, yeah, it made a lot of sense. It probably was that, that there's steric clashing between regions of neighboring fabs bound to the same subunits that were causing dissociation in the case of 31A as a fab only. And so Henry thought of an experiment that he might be able to do to kind of test this hypothesis. If E104 requires the full-length antibody because of the way that it's bound to the fabs, in opposite, say, of 31A, which is able to get steric clashing between the fab domains themselves, then maybe if we elongate the hinge a little bit, we would relieve the tension that is actually leading to dissociation, and the molecule would stop working. So in other words, if we elongate the hinge, we can achieve the same thing as if we clipped off, or if we clipped away the hinge altogether. Could this basically be the reason? A hinge plier. Well, the short story here is that, yeah, actually, that's exactly what we observed in the lab. As Henry made these hinge mutants, where he extended the length of the hinge, we were able to basically abrogate the effect of the full-length antibody. Sequentially, with longer and longer hinges, less and less and less inhibition. So there's actually a paper out on this right now, and it's really fascinating because there's a lot of different tools that came together to do this experiment. But the initial hydrogen exchange results actually suggested that this might be the case. I think this power, or that this story really demonstrates kind of some power of hydrogen exchange when used in combination with other methods. It led us to a hypothesis about the mechanism of action. It enabled a crystallization strategy for a project that had been trying to get a crystal for more than a year. And it agreed with all other structural methods, yet it took a lot less time. This was actually the first case where we demonstrated allosteric mechanism to be how an antibody in the G&E repertoire worked. And this was actually the first cryo-EM structure that Genentech ever produced. And it was of tryptase bound to the E104 V1 fabs. So because of all of this data and the epitopes, we decided to try to crystallize 4 fabs bound to the same tetramer in E104. And that eventually led to a high resolution crystal structure. But this just kind of shows you how the hydrogen exchange regions did really paint a good picture of the epitope. Last but not least, I'd like to share with you a story that is very different than the first two. It's on the other side of the spectrum. We're going to go into development now, and we're going to get back to protein folding, actually. I'd like to tell you a little bit about transportation and mechanical stress. Some of you might not realize it, but proteins are sensitive to mechanical stress, like simply shaking the solution that they're suspended in. You can see here a picture of two vials. One has been mechanically shaken, and the other is the control. You can see aggregates clearly have formed in the mechanically shaken sample. The control has nothing. How could hydrogen exchange tell us about this? Well, there's a really interesting paper written by a colleague of mine, two colleagues of mine at the time, Danielle Leesky and Ian Shee. And Ian was focused on this whole idea that it's all about the air-water interface, that the air-water interface leads to destabilizes an antibody, and that the air-water interface is basically coated with antibodies because it's hydrophobic. What he found was that in molecules that have, worse aggregation problems upon shaking stress, that they tend to have a greater surface pressure than antibodies that have a lower problem. And this surface pressure seemed to suggest to him that, perhaps there's even, some degree of unfolding that's happening at the interface that he was able to measure. Anyways, he came up with a metric where he said, okay, high change in surface excess with respect to time is going to cause an antibody to aggregate. It's going to be associated with antibody aggregation. And a low change in this pressure with respect to time is going to be a limited aggregation. This is a great tool for screening then to detect whether or not we're going to have a problem with our antibody. But it doesn't quite tell us why. Why does a certain antibody have more of a problem and another antibody have less of a problem with the same shaking stress under the same conditions? I should have shown that a little bit earlier, but that shows the correlation between the change in the pressure and the aggregation. You should check out this paper. It's really fascinating, the tools that they use to make these measurements. So Chamali was working with me. She's a graduate student in David Weiss's lab. And we had this hypothesis that basically at the air-water interface, some antibodies are unfolding faster than others. And the ones that are unfolding faster or more than others, then over time will start to aggregate. Under shaking stress, you're basically constantly turning over this interface over and over and over again. Antibody, the sample will repartition to the interface incredibly fast. And over time, more and more molecules are unfolded. Over time, they're not having time to refold, and so they're aggregating. That was our hypothesis. Now, if this is true, we should be able to test it with hydrogen exchange. So the experiment being shown over here on the right, we should be able to take the antibody, get it into D2O by gel filtration, and then compare an agitated sample to a non-agitated sample. Take time points, quench them, and measure them by mass spectrometry. We should hopefully be able to see the antibodies who have bigger aggregation problems are going to take in more deuterium, and the ones that have less are going to take in less. What you're seeing here is actually peptides from two different antibodies, MAB alpha and MAB beta in this case. I can't tell you the exact names of these antibodies, but what I can tell you is that this one, MAB beta, will be a lot worse than MAB alpha if we don't protect it with polysorbate. Polysorbate is something I'm not going to get too much into right now, but the idea with polysorbate is that it coats the air-water interface. It's amphiphilic, and so it's going to stick its hydrophobic tails up into the air, and it's going to basically mimic bulk water from the perspective of antibody, and so the antibody doesn't see the interface. Well, we found out that the effects that we were seeing, so we were getting increased exchange in MAB beta as compared to, say, MAB alpha. We're showing the largest change in signal here, the red peptide on the left and then the same red peptide to the right. you can see that basically MAB alpha, which gives us no change in turbidity at all, gives us only a very slight destabilization when it's agitated. And that both of these antibodies basically are fully controlled with polysorbate 20. Now, polysorbate 20 has blessings and curses. I mean, polysorbate 20 is in almost all products that are, almost all parenteral products, because, subvisible particles are really important. You don't want subvisible particles and things that you're injecting into people. But polysorbate can degrade and then lead to subvisible particles on its own. So what we would ideally be able to do is engineer a molecule to be resistant to these effects. And being able to see locally what is the first region to unfold at the interface might allow us to go in and make selective mutations in these regions of the molecule to make it less sensitive to mutation. What was interesting is that there was I think I showed you a third molecule back here. Yeah, MAB gamma right here. Sorry that I skipped over this quickly. But you can see MAB alpha is basically showing us no difference. MAB beta is showing us a little bit of gain in turbidity, whereas MAB gamma is really bad. So we looked at the difference between alpha and beta, but what about gamma? Why? I mean, is MAB gamma going to be that much worse than alpha and beta? Yes, and as it turns out, it's fascinating to me, somebody who's interested in the kinetics of hydrogen exchange and interested in hydrogen exchange theory. And I'm showing you, differences in what's called EX1 kinetics, where basically every unfolding reaction leads to a labeling reaction, versus EX2 kinetics over here on the left. And these are kind of details, but the point is here, the whole antibody lit up. This molecule is basically unfolding at the interface. Hardly any of it is unaffected. In conclusion, what it means is that, well, we can directly explain why MAB gamma is aggregating more than MAB alpha and beta. We can even show where it's unfolding worse. And this, hopefully in the future will give us a utility in protein engineering to potentially engineer out these liabilities from our products without actually having to add things like polysorbate to protect them. And so with that, I'd just like to acknowledge the people who were involved in the work that I was able to share with you this morning. The Triptase team over here on the left, Bob, Henry, Claudio, James, and Tang Xing. Check out these two manuscripts if you're interested in learning more. The mechanical stress study was done with Chamali Gamaj and her Ph.D. Professor David Weiss and University of Kansas, and Jasper Lin, Ann Woys, and Ian Shee. And then the hit to lead work was actually done, working a little bit with Chamali and this team here from BCP. And so with that, I'd like to thank you for the opportunity to present, and I look forward to talking with you in the open panel session.